Hyperthyroidism is caused by an overactive thyroid gland. Figure 14 shows a person with a normal thyroid gland and a person with hyperthyroidism. State one effect of hyperthyroidism on the thyroid gland. Well, you can have a look there. You can see that the thyroid gland has swollen. Another possible thing you might have written here is that it could increase the thyroxine production. The thyroid gland is part of the endocrine system, so that's C. Explain how negative feedback involving the thyroid gland controls metabolic rate. I'm just going to use this as a recap. So we have our normal thyroxine concentration in the blood. If that thyroxine concentration drops down or decreases, then TSH is secreted. So that's thyroid stimulating hormone from the pituitary gland. And then the TSH stimulates the thyroid gland to secrete thyroxine. We're going to increase the thyroxine concentration in the blood and it's going to return to normal. If the thyroxine concentration increases, then the pituitary gland will stop the production of TSH. And because there's no TSH, then the thyroid gland won't be stimulated to make any more thyroxine, so therefore the concentration will drop down. There you need to write your answer in. Low levels or concentration of thyroxine causes a hormone called TSH to be secreted by the pituitary gland. TSH stimulates the thyroid gland to secrete thyroxine, increasing the thyroxine concentration in the blood. If the thyroxine concentration increases too much, then this will inhibit or stop the production of TSH, so no thyroxine will be made or secreted. Explain how hormones control the menstrual cycle. So I'm just going to quickly go over this figure of 8 diagram that you've probably already seen before. We've got 3 parts in the menstrual cycle that are involved. We've got the pituitary gland, the ovary and the uterus lining. The pituitary gland secretes FSH into the blood. That targets the ovary. So an egg matures. Then we've got estrogen is secreted, and that will do a few things. One thing it will do is it will repair and thicken, or it will cause the uterus lining to repair and thicken. It will inhibit FSH, so you only get in one egg to mature. And also, as these estrogen concentration increases, LH is stimulated to be released by the pituitary gland. LH targets the ovary as well, and that causes the egg that's been maturing to ovulate, and then progesterone is secreted by what's left behind. Progesterone is responsible for maintaining the thickness of the uterus lining in case that ovulated egg gets fertilized, and it also inhibits FSH and LH. I want to show you that this structure here, so we've got an egg cell and it's surrounded by lots of little cells. That whole thing there is called a follicle. And when the egg is ovulated or released by the ovary, it will come out of that follicle and the egg will go into the fallopian tube. The little cells that were around the egg become what's known as the corpus luteum, and it's that secretes progesterone. So that's only going to make progesterone once the egg has been ovulated. After a while, the corpus luteum will degenerate, and that means the progesterone concentration will decrease. So if progesterone's job is to maintain the thickness of the uterus lining and inhibit FSH and LH, if the progesterone is no longer there, then the uterus lining can't be maintained. So we get a menstrual bleed and the FSH and LH are no longer inhibited. So that means FSH is free to start the cycle again. So this is a really good diagram to memorize in case you have a 6-marker question like this, but of course we're going to have to write the answer out properly. You can write it in bullet points if that's easier for you. So here's the answer. FSH is secreted by the pituitary gland into the blood. FSH targets the ovary and causes an egg to mature and estrogen to be secreted. Estrogen causes the uterus lining to repair and thicken. Estrogen also inhibits any more FSH from being made. As estrogen levels increase in the blood, it stimulates the pituitary gland to secrete a hormone called LH. LH targets the ovary and causes ovulation to occur. And ovulation is the release of the egg from the ovary. The corpus luteum that's left behind secretes the hormone progesterone. Progesterone helps to maintain the thickness of the uterus lining. It also inhibits FSH and LH from being made. And as the corpus luteum degenerates, progesterone levels will fall and FSH is no longer inhibited. So the cycle will start again. A person with kidney disease may need kidney dialysis treatment. Figure 15 shows an experiment to show how a dialysis membrane works. At the start of the experiment, we have got this dialysis tube containing a solution of starch and glucose, and that is in a beaker of water and dialysis membrane. And then we can see the experiment after an hour. We can see that some of those molecules have actually moved out of the tube and into that solution in the beaker. Describe the method that would be used to find out what is present in the solution in the beaker after 1 hour. So you know you've got starch and glucose in the tube. We know that some of the molecules are going to move out into the solution. So if we want to test this solution, the surrounding solution, for sugar, then we need to add Benedict's solution to a sample of that solution from the beaker. We need to heat this Benedict solution with some of the beaker solution in a water bath. You need to really emphasize the heating. If you don't, if you just say, oh, just put the solution in a water bath, that won't get you the marks. So heat this solution in a water bath, and if glucose is present, there will be a colour change. You cannot get a mark for just saying colour change, so you are going to have to tell the examiner what the colour change is from and to. So for example, well, we know that Benedict's solution is blue, so it's going to change from blue to green, or possibly orange or yellow or red. It depends on the concentration, but any of those colours would be fine. That's going to get you your 2 marks there for the sugar. Some of you will be thinking, well, the starch molecules are too big to pass out through this membrane. So therefore, some of you might think, well, we don't need to test for starch because it's too big, it hasn't moved out. But you really should, just to be on the safe side, check that solution for starch as well. And to do that, that's really simple. You just add iodine solution to the solutions, the sample you've taken from the beaker. And if you get a colour change from brown to a bluey black colour, then you know starch is present. So again, we've said what the colour change is. We haven't just said, oh yeah, there'll be a colour change. Explain how this experiment represents a simple model of kidney dialysis treatment. I think this is quite a hard question actually to think, what exactly is the question asking here? But what it wants is this idea that you understand the dialysis is acting like a cell membrane. So it is permeable. And if we are talking about the dialysis, you don't have to write this, it's why I've put it in brackets, but we're talking about cell membrane of the cells lining capillaries. So the dialysis tubing is acting like a cell membrane. So substances can move through this membrane by diffusion. And diffusion is a really key word, underlined, you've got to have said that. And then there was another mark going for from a high concentration to a low concentration, and then you just might as well pop the ending on there, down a concentration gradient. In this question, you will be assessed on using good English, organising information clearly and using specialist terms where appropriate. Homeostasis keeps conditions in the body relatively constant. The amount of water in the body is controlled by homeostasis. So they've used the word amount, which is pretty poor. So they're inviting you to use the correct terminology in your answer. Kidney function is controlled by a gland in the brain. And describe how the water content of the blood is controlled. So if the hypothalamus detects a decrease of the water concentration in the blood, it will send electrical impulses, not signals. Make sure it's electrical impulses to the pituitary gland, and this will secrete a hormone called ADH. ADH makes the collecting duct, or you are allowed to put kidney tubule there, more permeable, meaning that water can leave the tubule by osmosis and be reabsorbed back into the blood, therefore raising the water concentration back to normal. If there is an increase in water concentration in the blood, then we're going to need to remove this excess water from the blood and make sure that it goes into the urine. So the hypothalamus will stop the pituitary gland from secreting ADH, and that means the collecting ducts or the kidney tubules become less permeable to water. So that means less water is reabsorbed into the blood, meaning that there'll be more water in the urine, and therefore we've got rid of that extra water from the blood and we've maintained the correct water concentration. And maintaining the correct water concentration is an example of homeostasis brought about by negative feedback. Question 3. Figure 7 shows the brain. Which part of the brain becomes more active if a person balances on one leg instead of standing on two legs? So that is the cerebellum, and that is part A. Name the part of the brain that is responsible for making a decision. So you will have put cerebrum, and you could have put cerebral cortex there. In most MRI scanners, the person being scanned needs to stay completely still. A functional MRI or fMRI scanner allows a person to move while the scanner makes images of the person's brain activity. Suggest how the fMRI scanner could help to find out more about the brain damage a person has. Now, it's got move in the questions, so we're going to have to be a bit more specific than using that word. What we can do is we can ask a person to carry out a specific task while taking the scan, and then we can see which part of the brain is active or inactive. Then we can compare this to a person with brain damage so that we can see exactly where the damage is on our person we're scanning. And you can't do that with an original MRI scanner because people have to stay very still in those. So these moving ones are very handy. Describe how the brain receives information about light entering the eye. You should include the names of structures in your answer. Well, there are photoreceptors or cells that detect light, which are found on the retina at the back of the eye. Then light energy is going to be converted into electrical energy. And then this electrical energy in the form of impulses are going to pass along the sensory neuron in the optic nerve to the brain. Human activities affect the environment. Describe the reasons why deforestation takes place and the effects deforestation has on the environment. So again, we've got another 6 marker here. Just to let you know, examiners will read the whole question and they will get an idea of the logical flow. So this will be the same for the other 6 markers you've answered. Then we're looking for reasons for the deforestation and the effects of deforestation, and there needs to be quite a few examples of both of those in order to get up into the 6 mark category. Deforestation is the removal of a large number of trees for different reasons. The trees can be used for building materials and paper, and then the cleared land could be used for growing crops such as palm oil, or maybe to rear cattle or livestock, or the land may be used for mining. All of these can have a devastating effect on biodiversity. So what does that mean? The removal of a variety of different trees and replacement with a crop of one type reduces the plant diversity that was there, and in turn this will lead to a loss of specific food chains and habitats for other organisms. We could also lose potential sources of medicines. One of the main things that I really want you to include in these deforestation answers is this next bit here. So when you remove trees, you're reducing the photosynthesis that can take place. And as you know, carbon dioxide is needed to be taken into the plant so that it can carry out photosynthesis. If you're chopping the trees down, there's going to be less photosynthesis, meaning less carbon dioxide will be absorbed from the atmosphere, and therefore the CO2 is going to build up in the atmosphere and contribute to the greenhouse effect, and that can lead to global warming. If you burn the trees, that will release, because combustion does release CO2 into the atmosphere, that's going to further lead to global warming as well. And don't forget that when you remove trees, you're going to be removing roots that hold the soil together, so that can lead to soil erosion and landslides. And that covers quite a few reasons and quite a few effects, so that's going to get 6 marks. I do want you to remember that cutting trees down doesn't mean they just simply release carbon dioxide into the air. They have to be burned to do that. Okay, the reason why the CO2 is increasing is because they're not taking it in through photosynthesis and maybe they're being burned. The reaction time of 5 people was tested using a computer. These people were then given 100 centimetre cubed of a liquid to drink. Their reaction times were recorded 10 minutes after drinking the liquid, and figure 9 shows the results. So we've got 5 people, reaction time in seconds before drinking the liquid and then after, and the difference there. Calculate the mean difference in reaction time. Give your answer in milliseconds. The major thing you've got to look for here is the answer is in milliseconds. Look at the table. That reaction time has been recorded in seconds, so you are going to have to do a conversion at some point in this. It's really easy for you to realise how to work out the mean. So you add up all the differences, 0.035. Don't worry about the minus, so it accepts either way, that's fine. 0.035 divided by 5, because there are 5 people, and then that's going to give you 0.007 seconds. We times that by 1,000 and that gives us an answer of 7 milliseconds. And of course you might have put the negative in front and that will get a mark as well. The drinks manufacturer wants to advertise the effect of the drink on reaction time. The manufacturer needs to confirm the effect on reaction time by improving the investigation. Give 2 improvements the manufacturer would need to make to this investigation. So instead of just saying repeat, what's going to get you the mark is things like repeating. So test drink on more people. So you're explaining exactly what you're repeating, or more repeats on the same people. Or perhaps you could repeat this investigation but using different volumes of drink, so you're changing the volume, or repeat it using different time intervals. So instead of going after 10 minutes, you could go after 2, after 4, after 6, after 8, etc, etc, etc. ADH is a protein hormone made up of amino acids. Complete the sentences to explain the link between amino acids and proteins. So this is a bit tricky. Large molecules like proteins made of smaller molecules are called. So we're talking about a large molecule made up of lots of smaller molecules. That should be polymers in there. And then once you've got polymers, it should be easy to get the next bit. So the smaller molecules or amino acids are called monomers. Alcohol inhibits ADH production. Person A and B both drank one small 100 milliliter drink. Only one of the drinks was high in alcohol. Look at the diagram of the kidney tubule in person A and B after the drink. So if we have a look at person A, there's no arrows there on that collecting duct. So that means there's no water being reabsorbed back into the blood. Person B, there is water coming out of that tubule and then going back into the blood. So we then need to remember that if ADH makes the collecting duct more permeable, then more water will be reabsorbed into the blood. Okay, so ADH is going to happen with person B. So if ADH is inhibited, then the collecting duct is not going to be permeable and water won't be reabsorbed into the blood. So we're going to lose it down that collecting duct and into the bladder. So person A definitely has their ADH inhibited. What's the question again? How can you tell from the diagram that person A's drink contained alcohol? Because alcohol inhibits or stops ADH production, which means their collecting ducts will be less permeable to water, meaning that water is not reabsorbed into the blood. After the alcohol has been removed from person A's body, control mechanisms will correct the excess loss of water caused by the alcohol. Describe these control mechanisms. So the water concentration of the blood will be low, and this will be detected by the hypothalamus. The hypothalamus will stimulate the pituitary gland to secrete ADH, which will increase the permeability of the collecting duct so that more water can be reabsorbed into the blood, and that means less will be lost in the urine. This question is about homeostasis. Define the term homeostasis. That is the maintenance of internal conditions of a cell or body despite changes in the internal or external environment, so that the cells or enzymes can always work at their optimum. Name the hormone released if the blood glucose concentration falls too low. That is glucagon. You've got to get this spelling right because glucagon is so similar to glycogen. The examiner needs to know that you've understood glucagon is the hormone. 2 people were sent to hospital to find out if they have diabetes. This is the method used at the hospital. Do not eat or drink after midnight. This is called fasting. Measure blood glucose concentration at 9:30 a.m. Drink a glucose solution at 10 a.m. Measure the blood glucose concentration for the next 3 hours. Person A and person B have diabetes, so they both have diabetes. Describe how figure 4 shows that person B has diabetes. Now, because they both have diabetes, this question wants you to compare person B with the key here, the normal blood glucose concentration. You're not comparing it to person A because they have diabetes as well. Describe how figure 4 shows that person B has diabetes. There are 3 parts of the graph that you need to describe. You've got this flat area here, you've got a sudden increase, and you've got a slow decrease in blood glucose concentration. So their fasting blood glucose is higher than the normal range. So if you look at the graph, 8.2 is person B's blood glucose concentration when they're fasting. And if you compare that to normal fasting should be between 3.5 and 5.5. So already their blood glucose concentration is too high. After they've consumed the glucose drink at 10 a.m, their blood glucose concentration increased rapidly all the way up to around 13.2 millimoles per decimeter cubed. And you can see that that is above the normal peak of non-fasting. So if you look down at non-fasting is a range between 4 and 7.8. 13.2 is way above 7.8. And even after 3 hours, their blood glucose concentration didn't even return to normal levels, not even their own normal levels. Person A and person B had a test to measure the concentration of insulin in their blood when they were fasting. So person A has quite a high concentration of insulin and person B doesn't really have enough. And you look at the normal range, person A is above the normal range and person B is below the normal range. So we have actually got 2 different types of diabetes going on here. Suggest which type of diabetes person A and person B have. Give a reason for each answer. Person A has type 2 diabetes, and the reason is because the pancreas is still producing or secreting a lot of insulin, but the body cells cannot respond to it. You have to be really careful here not to just say the body can't respond to it. You had to put the word cells in there, so the body cells cannot respond. Person B has type 1 diabetes, and you could tell this because the pancreas is not producing enough insulin. It's important to keep the blood glucose concentration within narrow limits. A person eats a meal containing a lot of carbohydrate. This causes an increase in the person's blood glucose concentration. Explain how the hormones insulin and, that's in bold, glucagon, so you have to say both of them, control the person's blood glucose concentration after a meal. The pancreas detects an increase of glucose in the blood and secretes insulin. Insulin makes the membranes of cells, especially liver and muscle cells, more permeable to glucose, causing them to take in more glucose, right? So the permeability of the cells, that is a second mark there. If there is excess glucose that has not been used for respiration, that can be converted to glycogen and stored as glycogen in the liver and the muscle cells. So the liver and the muscle cells are the only place where you can get this conversion and storage of glycogen. if you've got glucose being taken into the cells, out of the blood into the cells, and you've got excess glucose being converted into glycogen, that is going to reduce the blood glucose concentration. The pancreas will secrete glucagon, which targets the liver cells. Glucagon causes glycogen to be converted back into glucose. This glucose can diffuse back into the blood, and that's going to raise the blood glucose concentration again. And why not? We could just add in, for luck, just in case there's a mark going for it. This is an example of homeostasis. So keeping that blood glucose concentration is stable despite eating or not eating. That's a very important example of homeostasis, and it's brought about by negative feedback. If the blood glucose concentration increases, there will be a response. Insulin will be secreted and bring that back down to normal. If there's a decrease, glucagon will be secreted and that will increase it back to normal. So always bringing it back to normal. Number 8, gardeners sometimes make compost heaps from dead plant material. The dead plants decay the compost heap. And we've got, this shows the compost heap. Okay, the thin layers of soil contain organisms that cause decay. Which 2 types of organism cause decay? So that is the bacteria and the fungi. It can't be grass, that doesn't make sense. Insects and worms, they do break down materials, but they do that because they eat it, they consume it, and they use inside of their bodies to break it down. So that is digesting and not decay. The rate of decay in the compost heap depends on several environmental factors. Explain how the rate of decay would be affected by an increase in oxygen concentration, a temperature increase between 5 to 25 degrees C. So it's not getting too hot, so we don't need to worry about denaturing or anything like that in this particular question. There's a mark going for saying both of these will increase the rate of decay. 1 mark, job done. An increase in oxygen is needed so that these decaying microorganisms can carry out aerobic respiration to release energy. And the increase in temperature will increase the rate of reaction inside the organism, such as increasing their respiration or their own enzyme activity. Give 1 environmental factor needed for decay. Do not refer to oxygen or temperature in your answer. So please don't put oxygen or temperature in your answer. What you need to think about is the wow. When things decay, think wow. Warmth, meaning temperature, O for oxygen, and then W for water. So that is your missing ingredient to the oxygen and temperature. We've got to have moisture involved there. You can't have any biological chemical reactions occur if they're not dissolved in water. Dead plant material can also be decayed in a biogas generator. Figure 2 shows the percentage of gases found in a sample of biogas. Gas X is the main fuel gas found in biogas. What is gas X? It's methane. What is the percentage of gas X in the biogas? So we've got our pie chart here. Let's take that whole circle as 100%. Carbon dioxide and other gases. So if you add those together, that gives you 40%. So you just got to work out the difference, which is going to be 60%. The dead plant material in the compost heap and biogas generator does not decay completely. Explain why a farmer might spread the remaining dead plant material onto his fields. Well, that is because the dead plant material can decay further, releasing more minerals into the soil, and that's going to help the plants to grow better. Number 9, diagram shows the carbon cycle. The concentration of carbon dioxide in the atmosphere has increased over the last 100 years. Give 2 human activities that might have caused this increase. The burning of fossil fuels is probably your most obvious one you're going to go with. We've already had a question on deforestation, so you know that. You may have put destruction of peat bogs as well, because that does release a lot of carbon dioxide into the atmosphere. Give the 2 letters of 2 arrows in the diagram which show respiration. If we're looking for respiration, we're looking for arrows that are going to be pointing towards where the carbon dioxide is in the atmosphere. Dead plants and animals, there seems to be a release of carbon dioxide from that. The microorganisms that are decaying the dead animals and plants. So B is actually microorganism respiration. D, plants, yeah, because plants carry out respiration. And animals carry out respiration as well. So we could go with B, C or D. So I'm just going to put B and C in mine. But you could put D, that will give you the mark. Give the letter of one arrow which shows decay. I already mentioned about the dead animals and plants being broken down and decayed by microorganisms. So that is going to be B. Scientists investigated the breakdown of dead leaves. The scientists placed dried leaves in mesh bags. So these are bags with small holes in them. Half of the bags had a mesh size of 1.5 millimeters. The others had a mesh size of 6 millimeters. The 6 millimeter mesh will have bigger holes in it, and the mesh with 1.5 millimeters will have smaller holes. The scientists then weighed the dried leaves in each bag at the start of the investigation. Placed the bags of leaves on soil. Some of the bags were placed in areas where there were earthworms in the soil, and the other bags were placed in areas where there were no earthworms. Left the bags for 4 months, collected the bags, dried the leaves and weighed them again. Most earthworms are between 3 millimeters and 6 millimeters in diameter, so that's quite handy to know, because if we look over at our mesh bags, most of these worms are going to be way too big to fit through the holes of the mesh size of 1.5 millimeters, so they're not going to get into this bag and eat the leaves. The 6 millimeter mesh is big enough for most of the worms to get in, so the worms can get into those bags and eat some of the leaves. The bar graph shows the scientist results. The percentage of leaf litter at the start of the investigation was 100% in each bag. What percentage of leaf litter was broken down in the 6 millimeter mesh bags? This is a bit of a horrible trick question, but I want you to get used to the habit of this anyway, is checking your labelling of axes. If you look at the y-axis, the percentage of leaf litter remaining is what is being shown in these bar charts. So it's very important that you've understood that in the 6 millimeter mesh with earthworms, you have 10% remaining. But the question is asking what percentage of the leaf litter was broken down. So you have to put 90% because that is the difference. Without earthworms, you're left with 22%. So that means that 78% has been broken down. So a common mistake here would be to just simply read 10 and 22 and put that as your answer. But it's just paying attention to detail and reading the question and checking your axes. What effect do earthworms have on the amount of leaves broken down in the 6 millimeter mesh bags? Use your answer to part BI to show how you arrive at your answer. Earthworms increase the leaves being broken down by a further 12%. So if you're looking at your 6 millimeter and you've got 90% with earthworms and 78% without earthworms, and there is that difference, isn't there? So the earthworms do make a difference by about 12%. When there were earthworms in the soil, the results for the 6 millimeter mesh bags were different from the results for the 1.5 millimeter mesh bags. They are using the word different. You're not allowed to say different. You need to say in which way they're different, whether increased or decreased, and give details. Explain why. The worms were too big to get into the 1.5 millimeter mesh bags, but could get through and into the 6 millimeter mesh bags. This enabled the worms to increase the breakdown of the leaves because they ate them. Other organisms smaller than earthworms cause most of the breakdown of the leaves. Explain how the results show this. Well, if you have a look at the results, you can see that the breakdown still occurs in mesh bags that are 1.5 millimeters. No earthworms can actually get into those bags. So that must mean that most of the breakdown does occur without the worms. It implies that other organisms are being used to decay. And that is the end of those questions. I hope you found the walkthrough useful. Good luck in your exams.